In this session, I will discuss about the importance of pentose phosphate pathway and that is also called as HMP shunt pathway, and roles of NADPH which is produced in this pathway. Objectives we have set in this pathway in this session are significance of HMP shunt pathway and various roles regarding NADPH. Let us first discuss about the significance of this pathway. Very important pathway, this particular pathway, significance. The first one is the production of NADPH. Okay. The second one is the provision for pentose sugars. What are these pentose sugars? The pentose sugars here, one of the pentose sugar which is produced in this one is ribose. Of course, other pentose sugars are produced. Then ribose can be converted to deoxyribose. Okay, deoxyribose that can be used in the synthesis of DNA, and ribose can be used in the synthesis of RNA. That means the nucleic acid synthesis, the pentose phosphate pathway will be important. Okay, the pentose phosphate pathway, when you have studied, you must have remembered that this pathway can be divided into oxidative pathway and non-oxidative pathway. In the oxidative pathway, as far as the oxidative pathway is concerned, so here there is a production of NADPH and then followed by the non-oxidative pathway where pentose sugars or iptose sugars, others, they are produced. Okay. Now, once this NADPH is produced, what are the various roles of, that is the second one, what are the various roles of NADPH which is produced? Okay. The first one which I will discuss about this one is NADPH for the reductive synthesis. Synthesis. In the reductive synthesis, what does it mean? See, there are oxidative metabolism and reductive metabolism. So where reduction is taking place in the oxidation, generally it is NADH that goes to the electron transport chain where ATP production is taking place, and whereas here the reducing the one substrate leading to the other one, reductive pathway. So what are these reductive synthesis processes? So for example, I can say fatty acid synthesis where NADPH is used. Okay. Then other one is the cholesterol synthesis. It is the synthesis of cholesterol where a large number of this NADPH is required. Large number of NADPH is required for the cholesterol synthesis. Okay. Then, so that is the reason, not only these, the NADPH is required for the steroid synthesis, other steroids. Okay. Then also the various other molecules it is required, various other reactions which is required. Okay. So that is the reason the HMP pathway is active in number of tissues. So let us see, it is active. Of course, it is very low in the, as far as this pathway is concerned, muscles, but so in other tissues it is very much active. Okay, let us see. So I will give the details of various tissues where this pathway is active. So ovaries, of course it is understood, there is a steroid synthesis. Then testes, also same thing. Okay, then adrenals, also same aspect. And lactating mammary gland. Okay, so then thyroid. Okay, then liver, not but the least RBC. Okay, so in various tissues this pathway is highly active. Okay, so that is about the reductive synthesis. The next we'll study about this one, the roles of the NADPH is free radical scavenging. Free radical scavenging. Okay, so removal of the free radicals. What are free radicals? So I will tell the various free radicals. Well, first, I would like to explain what is a free radical. Free radical is atom or molecule which is having unpaired electron. Okay, it is always looking for the other electron. That is the reason it is very active. One is superoxide radical. This is superoxide radical. Okay, then also there is hydroxyl radical. This is called hydroxyl radical. Okay, then apart from this one, non-radicals, it is H2O2. So where they are produced, when they are produced, when you take the cell, in the cell, various metabolic reactions which are taking place. This is the cell. In the cell, the metabolism is taking place. One or other metabolism is taking place. So their byproducts are formed. So other than the products, their byproducts are formed. Those byproducts can be free radicals, and also H2O2. H2O2. Then why we are discussing about these free radicals? The free radicals, so they are highly active. So they are known as oxidants. They are known as oxidants. These oxidants cause the damage. The oxidants cause the damage. So whom they damage? So DNA. Oxidants, known as oxidants, so damage. Okay, cause damage. These oxidants cause the damage. So damage the DNA. Okay, then the proteins can cause the damage to the proteins. Fatty acids, especially unsaturated fatty acids where double bonds are present, so they are prone for the fatty acid, this free radical attack. And other molecules, they can be damaged. So that is the reason. So the free radicals which are produced, they are to be neutralized. How they are neutralized? How they are neutralized? How they are neutralized? So this you can see here, there is superoxide radical. This is the superoxide radical. So we can say superoxide. Okay, then plus NADPH. So which one produced in the pentose phosphate pathway. Then what happens here? There is O2 is released. It is called superoxide dismutase. Superoxide dismutase. This is the enzyme. Then leads to formation of H2O2. H2O2 is less damaging comparison to the superoxide radical. Then again another molecule of NADPH plus H plus. Okay, then what is the enzyme here? The enzyme is peroxidase. The peroxidase will be acting on this one H2O2, leads to 2H2O. Harmless water is produced. This is the how this neutralization of the free radicals which is taking place. The next one is RBC membrane maintenance. RBC membrane integrity. Okay, integrity maintained, okay, by the presence of this NADPH. So what happens to RBC membrane? Why RBC membrane is prone to the damage? So let us see RBC, there is high amount of O2. Then high amount of O2 more prone to oxidant damage. So damage than other tissues, other cells. Why it is so? When there is high amount of this O2 is used, so there is a every chance of production of more free radicals that leads to the high amount of this damage. So increased oxidant species production. Okay, increased oxidant species production. Then what happens here? Peroxidation of what? Peroxidation of membrane phospholipids is taking place. Phospholipids, which are integral part of the membrane, so they maintain the structural integrity. Once the phospholipids damage is taking place, so what happens here? So there is leading to membrane damage. Membrane. So okay, damage is taking place. Okay, so then once the membrane damage is taking place, what happens? RBC can easily get hemolyzed. Okay, RBC can easily get hemolyzed. RBC. So what happens once the phospholipids are damaged? Then what happens? RBC can easily get damaged and hemolyzed. And then what happens? So then it leads to the hemolysis. Okay. Then this is the another aspect. Then the how this can be prevented, how RBC membrane integrity is maintained. How RBC membrane integrity is maintained. Let us see. Very interesting. Okay, how this NADPH is useful in this case. Let us see. Here, when I say take the example of H2O2, the H2O2 leads to formation of 2H2O. So by the action of enzyme glutathione peroxidase, it is the peroxidase enzyme. So it requires a trace element, that is selenium. So in the presence of whom? Let us see here. Okay, here the glutathione, reduced glutathione is converted to oxidized glutathione. Oxidized glutathione. What is glutathione? Glutathione, you must have studied in the section of the short peptides, biologically important peptides. It is a tripeptide containing gamma glutamyl, cysteine, glycine. It is present in either reduced form or oxidized form. Reduced form, it is active, it will be useful. Oxidized form, it is not useful. The oxidized form has to be converted back to reduced form. Then only it can be reused. So then the next aspect which comes is how this oxidized glutathione is converted to reduced glutathione. What is that enzyme? That enzyme is glutathione reductase. The enzyme is glutathione reductase enzyme. So which coenzyme is required? That is the question which I say. Which coenzyme is required? This coenzyme is NADPH plus H plus. So leads to formation of NADP plus. So in the presence of NADPH, oxidized glutathione is converted to reduced glutathione. The NADPH is regenerated through the HMP shunt pathway. So through the HMP shunt pathway, shunt pathway. That is the reason. Apart from the glycolysis in the RBC, a large amount of glucose is diverted to the HMP shunt pathway so that the membrane integrity can be maintained. Not only that one. In the RBC, the purpose of having NADPH, the purpose of having HMP shunt pathway is to keep hemoglobin in a proper form. Let us see in the next one. Okay, so here it is methemoglobin. It is about methemoglobin. Methemoglobin, to hemoglobin. Methemoglobin to hemoglobin. HMP shunt pathway indirectly is required through supplying the NADPH. Suppose in the hemoglobin, iron is ferrous state. This is if it is in the presence of oxidants or free radicals. What happens here? Free radicals. Then it converted to Fe3 plus. This is hemoglobin. This is methemoglobin. Okay, methemoglobin Fe3 plus is present. It cannot bind the oxygen. It cannot transport. Then what's the purpose of having this one? So the thing is, it leads to methemoglobinemia. If the levels are increased, that means it has to be converted back to hemoglobin by converting Fe3 plus into Fe2 plus. How this process is taking place? Let us see here. HB. Okay, Fe3 plus. This is methemoglobin. Okay, then it has to be converted to the hemoglobin. Hemoglobin having Fe2 plus. This is hemoglobin. Hemoglobin. Okay, so who does it? So there is an enzyme system containing cytochrome B, Fe2 plus. It is converted to cytochrome B5, cytochrome B5, Fe3 plus, Fe3 plus. So by the action of methemoglobin reductase, methemoglobin reductase. Okay, so this is the one which is taking place. Okay, this has to be converted back to this one Fe2 plus regeneration. Who is helping this one? This is helped by NADPH plus H plus and converted to NADP plus. But it is not entirely NADPH which is acting as a coenzyme. Only NADPH is 30%. 30% contribution. Remaining is NADH. NADH. Remaining is NADH. NAD is converted to NAD plus. Remaining 70% is NADH. Even then, 30% itself is a big amount as far as the RBC is concerned. That is the reason the pentose phosphate pathway in the RBC is very important. Okay, in the next one is the detoxification. Detoxification of drugs and other agents. Detoxification of drugs or medicines which we take. Okay, other agents. Suppose this is RH plus O2. Okay, metabolism of a drug is taking place. So that is, what is the enzyme system? Cytochrome P450 monooxygenase system. Okay, monooxygenase system. So here, what we require, what is the coenzyme? Of course, it is NADPH plus H plus converted to NADP plus. This comes from the HM patient pathway. So the drug is metabolized. Once the drug is metabolized, it can be excreted. The detoxification reaction. You study generally we study in the genobiotic section. So the harmful substance is converted to less harmful substance, so that it can be excreted. Mostly it takes place in liver. So drugs which are to be excreted out after the metabolism. So here there is a cytochrome P450 system is there. So monooxygenase enzyme is there. Using NADPH, it is metabolizing it so that these drugs, they can be other agents, they can be excreted out. Then the last one, not the least, okay, that is bactericidal action. Bactericidal activity. So activity, we talk in reference of macrophages. Okay. Here, what happens here? Here, what happens? Macrophage and other cytotoxic cells cytotoxic cells. So suppose example neutrophils. Okay, so what happens? These cells, so they use the free radicals, use the oxidants. So they using oxidant species. So then what they do? They kill pathogenic microbes. Okay. Then, so what it can be, how it can be explained? So bactericidal activity is a protective mechanism. Whenever they attack by the pathogenic organism, it has to be killed. This is done by using the free radicals, oxidant species. During that period, there is increased consumption of O2. So increased consumption of O2, then we say respiratory burst. Respiratory burst. Okay, so here, so they're using oxidant species. Free radicals are produced. Here, free radicals are used for the beneficial function of the cell. Okay, how it is being done? Let us see. O2 plus NADPH, NADPH plus H plus. Okay, then what is the enzyme? NADPH oxidase, NADPH oxidase. Okay, then here NADP is released. Then what happens? Production of superoxide radical. Once the superoxide radical is produced, then what happens here? Again NADPH plus H plus is used, converting NADP plus. What is that enzyme? The enzyme is SOD, superoxide dismutase, leads to formation of H2O2. This H2O2 in the presence of chloride, the enzyme is myeloperoxidase. Myeloperoxidase. So it is leading to the production of highly active SCLO, that is radical. So it is as bactericidal action. Bactericidal action, kill the pathogens. Okay, then so this is the how the using the free radicals cell is using for its own advantage in the macrophages and cytotoxic cells. I will summarize the whatever we talked about so far. Okay, very important aspect related to pentose phosphate pathway, also known as HMP shunt pathway, and also we have discussed about the rules of NADPH. For this session, we have selected two objectives. One is significance of HMP shunt pathway and various rules of NADPH. Other aspects related to HMP shunt like glucose 6-phosphate dehydrogenase and its deficiency and other aspects we will discuss in the next session. Okay, then I talked about the significance of this pathway. One is production of NADPH and provision for the pentose sugars. Pentose sugars, one of the pentose sugar is ribose. Ribose can be converted to deoxyribose. Deoxyribose is part of the DNA. Ribose is part of the RNA. That means pentose phosphate pathway will be useful for the synthesis of nucleic acids. Then second one, what we talked about the roles of NADPH. In the various roles of NADPH, we have studied total 6 roles. We have studied. You can see how extensively NADPH, which is used. Generally, we give a lot of importance to the NADH, but NADH for the energy. NADPH is very important. Is also equally important for many reactions. The first one is direct to synthesis. So, like fatty acid synthesis, cholesterol synthesis, this NADPH is required. Then that is the reason HMP pathway, HMP shunt pathway is reactive in many tissues, ovaries, okay, testes, adrenals, okay, lactating mammary gland, then thyroid, okay, then liver, then RBC, okay, then the next one, we talked about the second one, about the free radical scavenging, removal of the free radicals. We talked about the free radicals. We said that atom or molecule having unpaired electron, they are highly active. They can damage other cells. Such free radicals are superoxide radical, hydroxyl radical, and non-radical is H2O2. That is also their byproducts are released. Some of the byproducts are free radicals and H2O2. So okay, so these can cause the damage. So that is, we can say oxidants known as oxidants, they cause the damage. Okay, so damage DNA, damage proteins, so damage fatty acids and other molecules. We have studied in the DNA, in the repair and DNA errors. One of the damaging agent is free radical. Okay, oxidative agents. Okay, then now how they are neutralized. how they are neutralized. So here they can be neutralized in this way. The superoxide plus NADPH plus H plus by the action of superoxide dismutase, it is called SOD. Okay, it is containing the various trace elements containing the enzyme, leads to hydrogen peroxide. So in the presence of hydrogen peroxide, so NADPH is used, and then by the action of peroxidase leads to harmless water is produced. Then the third one is maintenance of RBC membrane integrity. Okay, then RBC uses high amount of oxygen. There will be high oxygen pressure is there, so it is more prone to oxygen damage than other cells. So oxidant species and peroxidation of membrane, oxidant species cause the peroxidation of membrane phospholipids. So then leading to the membrane damage, okay, leading to the membrane damage. Then RBC can easily get hemolyzed and hemolysis. So that is the reason, one of the reason for the hemolytic anemia. So by the action of this free radical action, so how it can be, integrity is maintained. For the maintenance of integrity, RBC contain glutathione can be present in reduced form and oxidized form. So what happens, it has to be in the reduced form to carry out its function. So H2O2, that is the damaging molecule, then by the action of glutathione peroxidase, the selenium containing enzyme H2 is released in the presence of reduced glutathione. It is converted to the oxidized glutathione. It has to be converted back to reduced glutathione by the action of glutathione reductase. So how it happens? In the presence of NADPH. So this NADPH is converted to NADP plus. So this is converted back to NADPH by the HMP shunt pathway. Then the fourth one is the conversion of methemoglobin to hemoglobin takes place. So again, it has to be converted back. The hemoglobin has to be converted back to the methemoglobin. So how it can take place? In the hemoglobin, iron is in ferrous state. In the methemoglobin, it is in the ferric state. So that methemoglobin cannot transport oxygen. This may be taking place due to the presence of free radicals. So here there is one enzyme system is there, cytochrome B5. The enzyme system, cytochrome B5 enzyme system is there. Okay, the cytochrome B5 containing enzyme system, that is methemoglobin reductase. Okay, it can stay present in Fe2 plus and Fe3 plus. Methemoglobin converted to hemoglobin. In this turn, the cytochrome B5 Fe2 plus is converted to cytochrome B5 Fe3 plus, and it has to be regenerated in the presence of NADPH. Okay, then it can be regenerated, but it is not entirely by the NADPH, only 30%. 70% is by the NADH. It is 70% by NADH. Okay, then even then that 30% itself is a significant amount. Then detoxification of drugs. So here, drugs which are metabolized mostly in the liver, so has to be detoxified, or other agents have to be detoxified, converting into harmless substances and shall be excreted out. The one enzyme system in this aspect, cytochrome P450 monooxygenase, it uses NADPH and converting to NADP plus. So drugs are metabolized and excreted. The finally, bactericidal activity of macrophages, very important. Macrophages, they deal with the pathogens and also cytotoxic cells, neutrophils. What happens? They use the oxidant species and to kill the pathogenic microbes. So in this condition, what happens? High oxygen is used, high amount of oxygen is used, high pressure of oxygen is used. In this condition, what happens? Free radicals are produced. These free radicals can kill the pathogens. They kill the pathogens. Okay. So this is in that condition, during that period of infection, during that period, then there is sudden increase in the usage of O2 by the cells. That is, we call it respiratory burst. Okay, then how it is taking place? What is the role of NADPH? O2 plus NADPH plus H plus. NADPH oxidase and NADP plus is released, leads to superoxide. The superoxide, by the action of superoxide dismutase, NADPH plus H plus is converted to NADP plus, leads to hydrogen peroxide. Hydrogen peroxide in the presence of myeloperoxidase enzyme myeloperoxidase enzyme in the presence of chloride leads to radicals. So they are highly active. So like hypochlorous and others, they are highly active. So they are bactericidal activity they have. Okay. In this session, we have discussed about the pentose phosphate pathway and various other aspects. And in the next session, we will be taking G6PD role, glucose 6-phosphate dehydrogenase role, and the deficiency, consequences of the deficiencies, and other aspects of pentose phosphate pathway, a clinical importance. Okay, thank you. Thank you very much.